Alright, so our next video on nucleic acids, and this is really just a rehash of what you have suffered through in the previous videos, and this is the primary sequence, and that is what we formally call this 5' to 3' end. That's kind of how we ended up the last video, right? We discussed this whole concept that there is a proper order to how we write a strand of nucleic acid. And we write the 5' end to the left and the 3' end to the right, as you can see in the slide. And then all of the bases are listed with one letter. So A, T, G's, and C's or U's go in the middle. All right, so with this 5' to 3' end, this is what we refer to as the primary sequence of the nucleic acid. Very similar to the primary sequence when we talked about amino acids and proteins, right? We said amino acids hook up and the order in which they hook up make the primary sequence of the protein. The same thing happens here. These heterogeneous bases with the sugars and the phosphates, those units begin to hook up. And when those units hook up, well, this is what's starting to create our polymer, and this is what we call the primary structure of the nucleic acid. So, just a piece of terminology that you should be aware of, um, and probably a test question that you're going to see as well. All right, so that's the primary sequence. Now, of course, if that's the primary sequence, then we need to kind of relate this primary sequence to a 3D shape because just writing it on a sheet of paper is not going to do us any good. We actually want to know what these things begin to look like in a 3D fashion, in a 3D environment. What begins to go on? What kind of shape does it take? How does it interact with other things? And that's exactly what this next slide or the next couple of slides will go through. And that's this 3D structure of nucleic acids. What you will find out is that this 3D structure was very hard to determine back in the day. And this 3D structure of a nucleic acid tended to give us some problems. We can maybe pinpoint some of the things that this would maybe take shape of, but never really could we concretely identify or conclude what exactly this 3D shape was like. So, what kind of shape do these things take off? Well, that's the story with Watson and Crick. So, you've all heard of Watson and Crick, and this was the mid 1950s. So, a lot of times when we go over scientists and when we go over these researchers, you're actually seeing that these are early 1800s, early 1900s, and or 1700s, and never really have we seen researchers in the 1950s before. So, again, this gives you an idea of how new and how fresh this field of chemistry is, the field of biochemistry is, and this field of biology is. So, there's still quite a few things that we don't know, but once again, we've made quite a bit of progress since Watson and Crick determined this 3D shape of a nucleic acid back in the 1950s. Okay, well, we say Watson and Crick. The problem, though, is that there were other people involved besides Watson and Crick that really aren't given any credit at all. And it's kind of a sad story, but it's a story worth mentioning. So, one of science's major discoveries are getting ready to happen. One of the most important things that happen in our lifetime, really, that we can remember. Some of us were born in 1950. I wasn't, but some of us were born in the 1950s. But in the 1950s, we get a very dynamic research that goes on between a couple of different people. Watson and Crick were two, but there were some others, which is why I'm getting ready to call this not only one of the most important discoveries, but also one of the most important soap operas that we can actually witness and look about how people were treated just in the 1950s alone. And the reason that I say this is because one of the third person that typically gets left out is someone by the name of Rosalind Franklin. And Rosalind Franklin never really gets incorporated into the Watson and Crick story, but Rosalind Franklin should. And Rosalind Franklin should actually be the first person that's named, because if it wasn't for Rosalind Franklin, they maybe would have a harder time determining the 3D structure of this DNA molecule. Okay, so here's Watson. At the time of this video recording, Watson is still living. So once again, this is something kind of fresh, right? We see the scientist, we see this portrait, or his picture, we see some information about him, and then his death, we see a question mark. So we're actually talking about real time now. And then not only that, but this picture is a color picture, and it's not a drawing, and it's not a portrait that's been painted by somebody. It's actually a picture taken by a camera. Wouldn't you know it? So here we get a very important individual in our lifetime, really, that made a very important discovery when it concerns the 3D shape and structure of DNA. So if we take a look, he was born in 1928. He was a biologist by trade. We're going to label him as the co-discoverer of DNA. You can kind of read other things about him if you want, but one of the ways that he determined the structure of 3D nucleic acid is using pieces of cardboard and an X-Acto knife. So one of the reasons that cardboard was used is because Watson was very familiar with the idea of planar molecules and these aromatic molecules. And you take a look at those purine and pyrimidine bases, and you're going to see some aromaticity to them. They are, for the most part, perfectly flat. And because they're flat, then we needed a flat surface to work off of that would not fold and be flimsy like a sheet of paper. So why not use some cardboard? So that's exactly what he did. So made the first 3D shapes of nucleotides by using cardboard and an X-Acto knife. He actually started his studies of birds and then changed it to genetics when he went to college. And he quotes, the cardboard shapes, since the bases are flat, allowed him to piece together information to see how they could fit together. And really what they were looking at here were hydrogen bonds. How can we maximize the number of hydrogen bonds, and how can we turn and twist this molecule in a particular shape in order for this hydrogen bond to take place at the maximum number that it can. Here is Crick. Crick, it is a black and white picture that's here. You could probably find a colored picture of him as well, but he did pass away in 2004. But still, again, 2004, that's our lifetime, really. So English biologist again. So we're starting to not really see chemist again, right? These are both biologists by trade that really made this breakthrough discovery. Coined the term central dogma, which means genetic information flows from DNA to RNA to proteins. That is the order in which your body begins to translate the message. So DNA converts the message over to RNA, and RNA converts the message over to proteins. And we will talk about that a little bit later on. But he is one of the ones that kind of coined that term central dogma to the whole field. PhD came from measuring viscosity of water at high temperatures. Yes, you read that right. We basically took water, increased it to a really high temperature, and measured the viscosity, or how thick it was, and did a research paper on it. Research happened during World War II. A bomb fell through the roof of his laboratory. It destroyed his apparatus. He had to start completely over. A lot of his data was lost and ruined, but the research went on. What a devoted soul. He occupied his time on how the brain makes a conscious mind, and he became a master of X-ray crystallography, which soon became the instrument that confirmed the DNA structure. Now, I want you to play a focus on that last statement. He became a master of X-ray crystallography. But during the time that this DNA structure was getting discovered, Watson and Crick didn't know anything about X-ray crystallography, at least how to run it and how to interpret it, because only a few people back during that time knew how to do this kind of work. And Watson and Crick were not specialists on X-ray crystallography during that time. So what basically happened is that they had these ideas, they had these insinuations of what these things looked like, and they needed proof. And they knew that an X-ray crystallographer could actually go in and give them the proof that they needed. But neither one of them were experts on X-ray crystallography. So they turned to experts in the field, and the experts in the field basically was a woman, and this woman was Rosalind Franklin. So Watson and Crick go to Rosalind Franklin, and they tell her, Rosalind, this is what we're thinking, and we need you to do the studies on them and let us know what you basically come up with. And she was a laboratory technician during the time, and she was a researcher during the time, and she knew how to run X-ray crystallography and how to interpret that as well. So she goes in and she begins to do these studies on them, and she has her laboratory notebook as well, and she begins to write things in the lab notebook, and she begins to write her data and her findings in the book itself, and then she begins to help out Watson and Crick in their discovery. Okay, well, I'll come back to that story in a minute, but this is what Watson and Crick proposed. These are the ideas that they have came up with to say this is what we think is going on with your DNA or your nucleic acid. So there's quite a bit here that you need to know and memorize. And yeah, I said memorize again, but there's quite a bit that you need to memorize. So in the next video, what we will do is that we will go through one bullet point at a time and talk about these in detail. So stay tuned. If you've got nothing to do, don't run away from me yet. Come back, take a look at the next video, and at least go through some of these things that Watson and Crick proposed.